In my lab, we study how the immune system functions. We want to understand how immune cells give us protection against infectious diseases and against pathogens that invade our body. Now, with this pandemic of the coronavirus, we want to understand, or we want to use our knowledge in order to create tools or new treatments to prevent this infection and provide protection against this virus or the disease that the virus is causing. In the blood of the patient, there are components of the immune response that basically provide them with protection or allow them to clear the pathogen and basically become healthy. And we are looking for these components. And the major components that we are looking for are antibodies. Antibodies that are specific for the coronavirus. How do we do that? How do we find antibodies against coronavirus in patients? So in my lab, together in collaboration with the lab of Ron Diskin at the Weizmann Institute, we produced a protein of the virus. It's a spike protein. It's presented on the surface of the virus. And basically, we take this protein and we try to fish out cells from patients that encounter coronavirus that can specifically bind this protein. These cells bind these proteins through antibodies. Now, if we fish out these cells, we can look at their genome and we can sequence the antibody encoding genes. And once we have sequences, we can basically produce the antibodies in the lab. And we don't need the patient anymore, or we don't need the cells that produce the antibody anymore. We can make the antibodies in large quantities in the lab. And this is a big advantage when we take antibodies from recovered patients. These antibodies were already selected. They were selected to attack the virus. They will not attack human tissues. They are not dangerous in any other way. And they cannot cause autoimmune diseases like in cases when you have antibodies that attack the body itself. So we are now at a very exciting stage. We took already immune cells from patients. We have the genome, or we have the sequences of the antibody genes. We are now producing them in the lab, and we are testing them for functionality and for binding of the virus. If the antibody has function, then we can suggest to use it in large amounts for treatment of patients. These antibodies can be like a drug and also like a vaccine, but these antibodies have to be tested in human beings, or in clinical trials, or in compassionate treatment of severe cases. And that will take about a year to figure out if the antibody is functional in human beings.